Welcome back, Trailblazers. You are listening to the Digital Pathology Podcast.
Glad to be here for this one.
Yeah, and we know you are all healthcare professionals, constantly pushing the boundaries of what is actually possible in medicine. So today, we're doing something a little bit special.
Right, we are doing a journal club-style deep dive.
Exactly, a deep dive into a piece of literature that is, quite frankly, going to fundamentally reshape the future of the operating room.
It really is.
Our mission today is to unpack a highly relevant, a systematic review published recently on February 20th, 2026. This is in the journal MDPI Diagnostics.
Authored by Chiara Innocenzi, Matteo Pavone, Barbara Seeliger, and a really incredible international team.
Yeah, it's a massive team effort. And the topic we are exploring today is that leap from standard image-guided surgery to real-time computer-assisted diagnosis using hyperspectral and multispectral imaging, specifically in gynecologic oncology.
And it is a remarkable synthesis of where we currently stand with this technology. Because the authors, they screen hundreds of articles to evaluate this exact transition.
And for the Trailblazers listening, the operational why here is just incredibly clear. I mean, we are all intimately familiar with the frustrations of the current gold standard.
The intraoperative frozen section.
Yes, the frozen section. You are in the middle of a procedure, you resect a margin, you send the sample down to pathology, and then you stand there and wait.
You're just watching the clock.
Literally. It eats up precious operative time. It requires really meticulous specimen preparation. And even then, we know it has suboptimal accuracy.
Especially when dealing with really complex margin assessments.
Right, or sentinel lymph node evaluations. We desperately need something faster, something that provides cellular-level insight while the tissue is actually still inside the patient. Okay, let's unpack this.
Let's do it.
Let's start with the fundamental science of how we are moving beyond standard surgical vision. Because we are all used to the limitations of standard RGB laparoscopic feeds.
The red, green, and blue.
Right. They only show us those RGB wavelengths, which completely restricts us to what the human eye can already see anyway.
And that limitation is the bottleneck of modern surgery, because biological tissue interacts with light in ways that our eyes and our standard OR monitors just completely miss.
They don't capture the full picture at all.
Not even close. When photons hit biological tissue, 4 distinct photophysical processes happen simultaneously. You have refraction, scattering, absorption, and fluorescence.
Okay, so 4 different things happening to the light all at once.
Exactly. And these interactions alter the speed, the direction, and the spectral composition of the light bouncing back. And here is the key: malignant tissues possess radically different morphological and biochemical properties compared to healthy tissues.
Because they're structured differently at a cellular level.
Right. They have disrupted collagen structures, dense and chaotic vascularization, altered cellular metabolism. And because of these structural differences, tumors interact with light differently.
They leave a different signature.
They create a highly specific spectral fingerprint. And this fingerprint extends across the entire electromagnetic spectrum, from the ultraviolet range all the way through the near-infrared.
So we capture this spectral fingerprint, but how does that actually translate to a screen in the OR? Because I saw the term hypercube repeatedly referenced in the paper.
Yes, the hypercube.
What exactly are we looking at when we talk about a hypercube?
Think of it as a massive 3-dimensional data set. So you have your 2 standard spatial dimensions, right? The X and the Y axis.
Like a normal photograph.
Exactly, representing the physical layout of the tissue. But then you add a 3rd dimension, the Z-axis, which is the spectral dimension. So instead of just 3 colors, hyperspectral imaging, or HSI, captures images in dozens or even hundreds of narrow, discrete bands. We are talking about slicing the light into intervals of maybe 10 nanometers across a huge range.
That is incredibly detailed.
It is. And multispectral imaging, or MSI, is kind of a cousin to this. It typically captures around 10 specific, slightly wider bands that are targeted for a very particular biochemical marker.
That sounds like an overwhelming amount of data to process.
It is a lot of data. But what's fascinating here is that this immense data density within the hypercube allows surgeons to effectively look inside the molecular structure of the tissue in real time.
Without contrasting agents.
0 contrast agents. Without physically touching the delicate tissue and without freezing and slicing it, you can quantify hemoglobin and deoxyhemoglobin concentrations right there.
Wow.
You can map out angiogenesis instantly. You can even assess the exact water, lipid, and protein content of a highly specific region of interest. It is, for all intents and purposes, an optical biopsy.
An optical biopsy. That concept alone could completely redefine surgical oncology. But to achieve that optical biopsy, we obviously need the right hardware.
Absolutely.
And the review breaks down 3 primary methods for capturing that light. And it seems to come down to a pretty brutal physical trade-off between spectral resolution, spatial resolution, and time.
The classic engineering triangle.
Right. So let's look at the first method they highlight, which is spatial scanning, sometimes called whisk broom or push broom scanning.
Right.
The paper notes this gives us incredibly high-resolution spectral information, but it gathers it pixel by pixel, or line by line.
And the mechanical reality of that process means it takes time. Gathering that data line by line can take several seconds to build the full hypercube.
Which in a laboratory setting is perfectly fine.
Sure, benchtop physics, it's great. But in an operating room, on a living, breathing patient whose organs are shifting with every respiration and heartbeat.
Several seconds is an eternity.
It really is. It makes the system highly susceptible to patient motion artifacts.
It sounds kind of like trying to take a high-definition panoramic photo on your phone while the subject is just walking around.
That is a perfect analogy.
One slight movement and the entire image gets distorted.
Exactly. If the tissue moves even a millimeter while the camera is scanning that specific line, the spectral data for that region no longer aligns with the spatial data.
So the hypercube is scrambled.
Your hypercube is essentially useless for a precise margin assessment at that point.
So spatial scanning gives us the detail, but it is just too slow for a breathing patient. The review then discusses the second method, which is spectral scanning. Does this actually solve the time issue, or does it just create a new problem?
Well, it attempts to solve the spatial problem by capturing the high-resolution spatial information of the entire surgical field all at once.
Okay.
And to get the spectral data, it utilizes tunable filters to switch wavelengths one by one. So the camera captures a full 2D image at 400 nanometers, then another at 410 nanometers, then 420, and so on.
But wait, wouldn't the time it takes to mechanically or electronically cycle through all those specific wavelengths mean you are still vulnerable to motion artifacts?
It absolutely does. While the spatial alignment of any single frame is perfect, if the patient breathes between the capture of the 400 nanometer frame and the 700 nanometer frame,
the z-axis is messed up.
Exactly. The z-axis of your hypercube is now corrupted. The algorithm won't be able to accurately compare the spectral signatures of a single pixel because that pixel represents a different piece of tissue in different frames.
Which brings us to the third hardware option they analyzed, which are snapshot devices.
Right.
These capture both the spatial and the spectral data simultaneously. It happens at video rate, meaning true real-time feedback for the surgeon.
Yes.
But the catch is that to achieve that kind of speed, you have to sacrifice a bit of both the spatial and the spectral resolution. You basically can't have the absolute highest definition in all 3 dimensions instantly.
It is a balancing act of physics. However, snapshot technology is advancing incredibly fast. And for many clinical applications, the resolution they can achieve at video rate is actually proving to be more than sufficient to distinguish between healthy and malignant tissue.
Well, let's look at what actually happens when you get this technology into the clinic, because the data here is staggering.
It really is.
The review screened 230 articles and narrowed it down to 29 studies and 2 clinical trials that met their really rigorous inclusion criteria.
And the bulk of the research right now is heavily focused on cervical neoplasia.
Yeah, making up almost 59% of the studies, and then ovarian cancer made up about 24%.
And when the authors aggregated the overall performance metrics comparing the spectral imaging readouts against the final histological pathology, the true gold standard,
the results were broad but impressive.
the overall sensitivity ranged from 75 to 100%, and the specificity ranged from 30 to 99%.
Those are pretty wide ranges.
They are, but when you drill down into the specific tissue types and the specific technologies used, the clinical implications become very compelling.
So let's focus on cervical neoplasia first. The standard approach right now is a pap smear, cytology, a conventional colposcopy, and targeted biopsies.
Right. Standard of care.
And we all know that process relies heavily on the clinician's visual experience, which leads to significant inter-observer variability.
Exactly.
In one comparative analysis highlighted in the review, hyperspectral imaging achieved a 97% sensitivity for detecting high-grade lesions, specifically CIN2 or greater.
Which is fantastic.
To put that in perspective for you trailblazers, the standard pap smear in that exact same comparison only managed a 72% sensitivity.
And the true power of this technology emerges when you look at how it tracks dynamic biological reactions.
What do you mean by dynamic?
So during a routine colposcopy, clinicians apply acetic acid, which temporarily alters the light-scattering properties of precancerous tissue.
Right, it turns white.
To the naked human eye, it just looks like the tissue turns white. But with multispectral imaging, you can continuously track the precise temporal pattern of that tissue whitening across the spectrum.
So it's measuring exactly how it whitens over time.
Yes. The review highlighted a fascinating study demonstrating that high-grade CIN3 lesions exhibit significantly prolonged whitening, taking over 500 seconds to relax back to their baseline state.
Over 500 seconds.
Right. But a lower-grade CIN2 lesion takes only about 200 seconds.
So you are not just looking at a static snapshot anymore.
Yeah.
You are quantifying a dynamic cellular reaction in real time, which completely removes the subjective guesswork of how white a lesion appears to a specific doctor on a specific Tuesday.
Exactly.
And the results of that quantification are phenomenal. The review noted that using MSI colposcopy drops the false diagnostic rate down to an astonishing 1.7%.
It's incredible.
When you contrast that 1.7% with a false diagnostic rate of over 20% for conventional colposcopies and over 24% for pap smears, that represents just an incredible reduction in patient anxiety, misdiagnoses, and unnecessary invasive biopsies.
The clinical relief there cannot be overstated. However, the narrative becomes far more complicated when the authors shift their focus to ovarian cancer.
Right, let's talk about the ovaries.
The studies examining ovarian tissue also demonstrated incredibly high sensitivity, often ranging from 81 to 100%. One study utilized a multispectral spatial frequency domain imaging system, or SFDI.
SFDI, okay.
This involves projecting specific structured light patterns onto the tissue to separate the effects of scattering from absorption. Using this, they found malignant ovaries had significantly higher total hemoglobin and reduced scattering amplitude.
Which aligns perfectly with the dense angiogenesis we expect to see in really aggressive tumors. But there is a snag with ovarian cancer detection that our trailblazers need to be aware of. While the sensitivity was phenomenal, the specificity numbers were highly variable and sometimes quite low.
This raises an important question about spatial heterogeneity within the abdominal cavity. Ovarian tumors are notoriously complex. You have hypervascular regions, you have pooling surface blood, areas of necrosis.
It's a messy environment.
Very. And blood acts as a massive optical sink. It absorbs huge amounts of light, heavily confusing the optical signals bouncing back to the sensor.
So it throws the whole reading off.
Yes. In one autofluorescence study the authors reviewed, this spatial heterogeneity caused benign endometriomas to be completely misclassified as malignant. It dragged the specificity down to a concerning 51.1%.
That is a lot of false positives that could lead to overly aggressive resections of completely benign tissue.
It is. But the researchers were able to implement mathematical normalization techniques, specifically a reflectance-based division method, to actively correct for that optical heterogeneity.
They fixed it in the software. Essentially, yes. By mathematically compensating for the pooling blood, they pulled the specificity up to 68.5% without hurting the sensitivity.
So it proves the data is actually there.
Yes, but dealing with the messy reality of surgical blood remains a major engineering hurdle.
Well, before we leave the adnexa, there is a fascinating development the authors included regarding the fallopian tubes.
Oh, the falloposcope.
Yes. We know from recent pathology that most high-grade serous and ovarian cancers don't actually start in the ovary. They originate in the salpingeal lumen of the fallopian tube.
Right.
So the review detailed a feasibility study for a device they're calling a falloposcope. It combines multispectral imaging with optical coherence tomography, or OCT, which is essentially like ultrasound, but uses light instead of sound waves for ultra-high-resolution depth imaging.
It's brilliant.
During a standard of care surgery, they were actually able to safely navigate this microendoscope into the incredibly narrow fallopian tubes in vivo to capture histology-like images of the tubal epithelium. The potential for early detection and surveillance there is just staggering.
Truly.
And it represents a massive engineering feat to miniaturize those sensors. But as the review makes clear, capturing this incredible data is really only half the battle.
Right, because of what we talked about earlier.
Exactly. Whether you are using a microscopic falloposcope, a snapshot camera, or a bulky spatial scanner, you are generating a hypercube. And that raw, multi-dimensional hypercube data is completely unreadable to the naked human eye.
It's just a wall of numbers.
It is an overwhelming flood of numerical data spanning wavelengths we cannot perceive.
Here's where it gets really interesting, because to translate that massive block of invisible spectral data into actionable, real-time surgical decision-making, we need artificial intelligence.
Yeah, I've had it.
The review found that nearly 45% of the included studies relied on some form of AI-based algorithm to interpret the hypercube.
And the majority of the researchers are currently relying on classical machine learning models. They are utilizing k-means, clustering, to group pixels with similar spectral signatures. They use linear discriminant analysis, support vector machines, and random forests.
Random forests essentially use a multitude of decision trees to basically vote on whether a pixel represents healthy or malignant tissue, right?
Exactly. However, a few of the more recent studies are pushing into deep learning, utilizing convolutional neural networks, or CNNs.
Now, CNNs are the same type of visual recognition architecture that allows a self-driving car to distinguish a pedestrian from a stop sign, correct?
In essence, yes. Instead of just looking at the spectral data of a single isolated pixel, a CNN looks at patches of pixels. This allows it to understand the spatial context, the textures, and the shapes of the tissue alongside the spectral data.
So it sees the forest and the trees.
Exactly. In one study comparing the two approaches, the CNN achieves significantly higher tissue type segmentation accuracy than a random forest model, simply because it could utilize a broader, more contextual range of the complex spatial and spectral information.
But the authors raise a massive red flag here regarding the black box problem, which is obviously a major concern for us in healthcare.
It's a huge issue.
In these early developmental stages, a lot of these studies are working with relatively small, unbalanced data sets. And when you feed a small data set into a really powerful AI model like a CNN, the algorithm is prone to learning completely spurious correlations.
And a spurious correlation in this clinical context is highly dangerous.
Because it's learning the wrong lesson.
Exactly. The AI might perfectly associate a diagnostic label like malignant with an optical artifact. It might learn that a specific glare from the operating room lights or a unique reflection off a metallic surgical instrument always appears in the cancer images. It then uses that glare to diagnose the tissue, rather than tracking the actual biomolecular tumor markers it is supposed to be analyzing.
So the machine is getting the right answer in the lab for the completely wrong reason.
Yes.
If you deploy that same algorithm into a different hospital with different overhead lighting or different retractors, it fails completely.
If we connect this to the bigger picture, it underscores the absolute necessity of explainable AI, or XAI, in the regulation of medical devices.
We can't just blindly trust the machine.
As clinicians, we cannot base an irreversible surgical resection on a black box algorithm that simply spits out a red or green heat map. Surgeons need to know why the AI is flagging that specific tissue.
Right.
We need to verify that the mathematical decision the model is making is biologically and clinically plausible. We need the system to say, I am flagging this region because the spectral signature indicates a 40% increase in deoxyhemoglobin coupled with altered collagen scattering.
So the surgeon can actually validate the physics against their own anatomical knowledge?
Exactly. It has to be a partnership.
Which brings us to the reality check. Because we always want to ground these incredible advancements for the trailblazers listening. Where is this technology actually sitting right now in the clinical pipeline?
That's the million-dollar question.
The authors mapped these studies onto the ideal framework for surgical innovation that tracks idea, development, exploration, assessment, and long-term study.
And they evaluated their technology readiness levels, or TRL.
Right. And the reality is this tech is largely sitting between TRL 4 and 7. It is firmly in the developmental and exploratory stages, specifically phases I and I.
We have fantastic lab validation and early clinical feasibility.
But we do not have the massive multi-center randomized controlled trials yet that prove widespread clinical effectiveness against current standards of care.
And the physical roadblocks in the operating room are significant. We discuss blood acting as an optical sink and confusing the ovarian signals, but surgeons also have to contend with cervical mucus, bile, and uncalibrated shifting operating room lighting interfering with the spectral capture.
It's not a controlled lab environment.
Not at all. Furthermore, for this to become truly ubiquitous in modern gynecologic oncology, we have to solve the miniaturization problem.
Getting it into the tools.
We need to figure out how to pack these highly sensitive, high-resolution hyperspectral sensors into minimally invasive 10mm laparoscopic and robotic tools without degrading the optical signal.
It is a tall order for the engineers, for sure, but the clinical momentum highlighted in this review is really undeniable.
It's moving very fast.
So, to summarize the landscape we have covered today, hyperspectral and multispectral imaging are effectively functioning as real-time optical biopsies. By capturing the unique, invisible spectral fingerprints of tissue, measuring everything from angiogenesis to lipid content, they have demonstrated incredible sensitivity for detecting cervical and ovarian malignancies.
And when eventually paired with robust, explainable AI, this technology could fundamentally reshape the concept of the smart hybrid operating room.
It offers the promise of giving you, the surgeon, molecular-level vision in real time, drastically improving complete resection rates and ultimately long-term patient outcomes. So what does this all mean?
It signifies that we are standing on the precipice of a new era in surgical precision. We are actively moving from a paradigm that relies solely on tactile feedback and the limited visible spectrum of human vision toward seamlessly integrating objective molecular signatures into our immediate surgical decision-making.
And that leaves us with a lingering thought for all the trailblazers out there. As these optical biopsies and explainable AI algorithms become rigorously validated and integrated into your standard surgical workflow, how will the definition of surgical intuition evolve?
That is a profound question.
If a machine can definitively see the microscopic molecular margin of a tumor that your human eye cannot, will future surgeons learn to trust the hypercube more than their own lifetime of anatomical experience? Where exactly does human judgment end and the algorithm begin?
It's something everyone in the field needs to be thinking about.
It really is a philosophical and clinical boundary we are all going to have to navigate very soon. Thank you for joining us for this Journal Club deep dive on the Digital Pathology podcast. Keep pushing the boundaries and keep questioning the future of medicine. We will see you next time.